Let's begin with the second chapter, that is sexual reproduction in flowering plants. Now in this chapter we are going to deal with the events like microsporogenesis, megasporogenesis, embryogenesis, seed formation, pollination and fertilization. These are the main terms that we are going to deal with in this chapter. Now when I am talking about sexual reproduction in flowering plants, before we start with the chapter completely, we need to learn about the flower properly. Yes or no? Flower. It has four main parts. Now which are the main parts in the flower? The first one is calyx, which you can also say it as a sepal. The second one is corolla, which you can also say it as petal. The third one is androecium, which is the male part, that is a stamen. When I say gynoecium, the fourth part, that is a female part, which is known as a pistil or a carpel. Right, let's go into detail about all the four components of the flower, all the four main parts of the flower. The first one is calyx, which you can also say it as a sepal. The second one is corolla, which you can also say it as petal. The third one is androecium, which is the male part, that is a stamen. When I say gynoecium, the fourth part, that is a female part, which is known as a pistil or a carpel. Right, let's go into detail about all the four components of the flower, all the four main parts of the flower. The first one that I said is calyx. Calyx is the outermost protective wall of the floral appendages. Right, now when I say it is the outermost protective wall of the floral appendages, it is an individual appendage of calyx is termed as what? It is termed as a sepal. Right, and that is the reason we say calyx is also termed as what? Sepal. Right, now what is the color of the calyx? It is green in color. The second part that we have to say is what? The second one is corolla. Corolla, which is also termed as petal. Now same as that of a sepal, that I can say the individual part. Now individual appendage of corolla is termed as what? The petals. Right, now it arises from the inner side of the calyx. Now as calyx is green in color, corolla, the petals are different, they are different colors, right? They are very attractive as well. Attractive in means of fragrance, it gives fragrance. Now when it gives fragrance, ultimately it helps where? In pollination. What is pollination? Transfer of pollen grains from anther to stigma, right? Anther, the male part, stigma, the female part of the flower. The next one is the androecium, which is the male part, that is stamen. Now it consists of stamen, the male part of the whole, right? Now when I say stamen, it involves two different parts. One is what? Anther. Second one is what? Filament. When I say anther, it produces the microspore, that is what? Pollen grains, right? And second one, filament, is what? It acts as a stalk of anther. Which one? The filament. Filament act as a stalk of anther. The next last part of the flower is what? The gynoecium. Now gynoecium, when I talk about it, is the pistil or the carpel. The individual gynoecium part, appendages of gynoecium, you can say it as a pistil or a carpel. Now this consists of stigma, style and ovary. It helps in production of megaspore, fruits and seeds. Let's begin with the next topic, that is pre-fertilization events and structures. When I talk about the pre-fertilization events and structures, we already know that the flower has the male part and the female part, that is androecium and the gynoecium. Androecium, which is a male part, gynoecium, which is a female part, which further develops and differentiates into to form what? The male gametophyte and the female gametophyte. Now when I talk about the development of the male gametophyte, we have to talk about the stamen. Now stamen has two parts, one is anther and one is filament. Anther that produces the pollen grain, whereas filament, which is a long slender stalk of anther, right? Now structure of anther is how? It is bilobed in structure with two theca. Now when I say theca, means what? It is a layers, twin layer associated. So it is bilobe consisting of two theca, or you can say it as what? Dithecous. I repeat, dithecous. For two theca, we say it as dithecous. So anther is bilobe consisting of two theca, or you can say it as dithecous, right? As you can see in the diagram, in the cross section, the anther is four-sided tetragonal structure. This tetragonal structure has a microsporangia in each corner, which is located in the corner. Now each microsporangia further develops to form pollen sac, which produces what? The pollen grain, right? Now when I talk about the structure of microsporangia, microsporangia has four different layers associated, which are these. The first one is epidermis, the second one is endothecium, the third one is middle layer, the last one is tapetum. The first three layers, that are epidermis, endothelium and the middle layer, they are the outermost layer, right? And tapetum is the innermost layer. When I say the outermost layer, the function is what? Protection. And also it prevents from what? It helps in dehiscence. It also helps in dehiscence of what? Anther to release pollen grains. The innermost layer, that is the tapetum. Tapetum nourishes the developing microspore or pollen grain. When anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Now when I talk about microsporogenesis, each cell of the sporogenous tissue is pollen mother cell, which we also say it as PMC or microspore mother cell, and can give rise to microspore tetrad. This process of formation of microspore from a pollen mother cell through the process of meiosis is called microsporogenesis. I repeat, the process of formation of microspore from a pollen mother cell through process of meiosis is called microsporogenesis. As anther mature, the microspore dissociates from one another and forms a tetrad and develop into a pollen grain. Inside each microsporangium, several thousands of microspores or pollen grains are formed. Now let's talk about the male gametophyte, that is the pollen grain. A pollen grain is a partly germinated microspore representing the male gametophyte. It is a haploid, uninucleate and minute spore produced in large numbers by meiosis in the microspore mother cell, that is MMC. They vary in their size, shape, color, design, etc. from species to species. When I talk about pollen grains, they are spherical in shape. They are almost 25 to 50 micrometer in diameter. It has two wall layers. That layers are termed as sporoderm. Two layers are termed as sporoderm. Now the names of the layers are what? The exine and the intine. The exine is the outermost layer and the intine is the innermost layer. Exine is composed of sporopollenin. I repeat, exine is composed of sporopollenin, which is a resistant organic material as it can withstand any high temperature, or any strong acids and even alkaline. There is no known enzyme which can degrade this, right? The second one is what? The intine, which is the innermost layer. It is composed of cellulose and pectin. As you can see in the diagram of a mature pollen grains, which has two cells, that is vegetative cell and the generative cells. Each mature pollen grain in angiosperms has two cells, the generative as well as the vegetative cell. The vegetative cell is larger, has abundant reserve food material and large nucleus, which is irregular in shape, whereas the generative cell is small, spindle shaped and floats in the cytoplasm of the vegetative cell. Pollen grains are generally shed at this two-celled stage in over 60% of angiosperms. In the remaining species, the generative cell divides mitotically and gives rise to two male gametes before pollen grains are shed. That is in the three-celled stage, you can see, right? Then we talk about this. Pollen grains of several plant species may cause severe allergies, bronchial infections leading to asthma, bronchitis, etc. Okay, now if you have to say something else, like pollen grains are rich in nutrients as well. In Western countries, large number of pollen products in the form of tablets and syrups are used to increase the performance of athletes and race horses. Now this pollen depends on the temperature as well, temperature and the humidity where it has to be stored. So it varies from species to species. Like for example, you can say 30 minutes in case of wheat, rice, and several months in case of families like Solanaceae, etc. Now once pollen grains are shed, they have to land on the stigma before they lose viability if they have to bring about fertilization. It is possible to store pollen grains for use in liquid nitrogen, that is a temperature say like minus 196 degrees Celsius, in pollen banks for later use in plant breeding programs. Now that I talk about the development of the female gametophyte, we have already spoken about development of the male gametophyte, that is stamen, which consists of anther and filament. Now let's talk about the female gametophyte. Now when I say about female gametophyte is about what I am telling about, it is a pistil or the carpel, which consists of three important parts, which are these: the stigma, style and the ovary. According to the diagram that we have drawn initially as well, that is, stigma is the structure which is associated to accept the pollen grain, meaning what, it acts as a receptive organ which accepts the pollen grain. The style is an elongated part of stigma, right, which helps the pollen grain to move towards the ovary. Ovary is a bulged, swelling part which consists of ovules. The pistil or the gynoecium, which represents the female gametophyte, it can produce one carpel, two carpel. Now when it is one carpel, it is known as monocarpellary. When I say two, it is bicarpellary. When I say three, it is tricarpellary. When I say many or multiple, you can say it as multicarpellary. Pistil may be syncarpous, that is, more than one pistil are fused together, right? As you can say solanum, etc. Or maybe apocarpous, that is, carpel remains free, as you can say in any rose, etc. Right? A typical pistil consists of a hollow basal swelling ovary, an elongated style and a terminal stigma. Stigma which serves as a landing platform for the pollen grain. As I have already told, it is a receptive organ which accepts the pollen. Inside the ovary, in the ovarian cavity, that is a locule, you can say, in which placenta is located. Megasporangia, that is the ovules, you can say, arise from placenta. If microspore is produced, that is microspore which is acting as a pollen grain, and megaspore, it acts as an ovule, right, which is an ovule. Megasporangia arise from placenta. Now when I have to talk about the structure of megasporangium or the ovule is an integumented megasporangium within which the meiosis and megaspore formation takes place. It is attached to the placenta by means of a stalk called the funicle. It is already given in the diagram. It develops into a seed after fertilization. The junction between an ovule and the funicle is called a hilum, right? Hilum is a junction between what? The ovule and the funicle, which later becomes a scar on the seed. Now each ovule here has one or two protective envelopes. Yes, that is known as integuments. What is it known as? Integuments. Now what does this do? It encircles the ovule except at the tip where a small opening called the micropyle is located. Okay, what does an integument do? It encircles the ovule except the tip where a small opening called the micropyle is located. The basal part of an ovule just opposite to the micropyle is called the chaliza. Enclosed within the integument in a mass of cells called nucellus, right? In the integument itself, there is a cell, there are many cells, mass of cells, meaning many cells, which is called as nucellus. The cells of the nucellus are rich in reserved food materials. Now an ovule which is there, it generally has single embryo sac, or you can say it as a female gametophyte, developed from a megaspore through reductional division and located within the nucellus. Let's move ahead with the next topic, that is megasporogenesis. Now when I talk about megasporogenesis, it is a process of formation of a megaspore from a megaspore mother cell, that is a diploid. It is known as what? Megasporogenesis. We already came across microsporogenesis as well with the same definition. What is the difference here? That is the pollen grains are added here. For example, the formation of what? Microspore from a pollen mother cell is known as what? The microsporogenesis. In the same way, megasporogenesis is the formation of a megaspore from a megaspore mother cell is known as megasporogenesis. Now here, it occurs inside the nucellus of developing ovule of angiosperm. The process begins very clearly when nucellus is not completely surrounded by the integuments. The MMC, yes, the MMC, a large cell. Now MMC is what? The megaspore mother cell. A large cell containing dense cytoplasm and a nucleus, prominent nucleus you can say, enlarges in size and divides by meiosis. It first divides transversely into two cells called megaspore diad. These two cells again divide transversely as a result of getting called a structure which is known as megaspore tetrad. What is it known as? Megaspore tetrad. Now when I have to talk about the female gametophyte, that is the embryo sac. In general, the development of an embryo sac is monosporic. Okay. Now monosporic, in this you can take an example of polygonum. In this type of development, only one megaspore situated towards chaliceal end, as you can remember through the diagram which we already studied earlier, remains functional, while the remaining 3 megaspore gradually degenerate and finally disappear. Only one remains. What is that? It is a chaliceal end, and the rest, only one remains, meaning what? Only one is functional, whereas the other 3 which is there, it degenerates and disappears. Following are the different stages in the development of a female gametophyte. Now, which are these? The first one, the functional haploid megaspore is the first cell of female gametophyte of angiosperm. It enlarges in size to form a female gametophyte, also called embryo sac. Its nucleus undergoes mitotic division to form 2 nuclei that move to opposite poles, forming 2 nucleate embryo sac. Now this 2 nucleate embryo sac, what does it do? It undergoes mitotic division. 2 more mitotic division. Now it undergoes 2 more mitotic division, means it doubles, that it becomes from 2 nucleate it becomes to 4 nucleate stage. And then again it doubles, later it becomes to 8. So 2 to 4, 4 to 8. That is the stage of an embryo sac that I am talking about. So this stage comprises of a micropylar end. That is the 8 nucleated stage I am talking about. It is termed as, it comprises of a micropylar end and a chaliceal end with 4 nuclei at each end. Yes or no? Because 8 nuclei. So 4 nuclei at each end. Now 6 of the 8 nuclei are surrounded by the cell walls and get organized into cells. 3 cells present towards the micropylar end grouped together constitute the egg apparatus. That says what? That is composed of 2 synergids and 1 is egg cell. Now 3 cells of chaliceal end are called the antipodals. The large central cell is formed by the fusion of 2 polar nuclei. Thus, a typical angiospermic embryo sac or a female gametophyte at maturity consists of how many nuclei? 8 nuclei and how many cells? 7 cells. This all for today. I hope you have understood this concept of female and male gametophyte. I will continue in the next video lecture. Thank you.